Hi, I'm Jackie.
And Jackie is going to help us today learn about pouring an agarose gel. So here we have a flask that has 50 mils of the TAE buffer with the appropriate amount of agarose in it. And Jackie's going to microwave that.
With the paper towel.
Yes, so you can also use a small glass flask upside down, but I always just use a paper towel. It works just as well. So then we're going to do time cook 45 seconds, and you want to not use maximum power or it'll kind of boil over. So you can hit start and then press power level 5 so that it doesn't run at full power. So when it's done, you want to remember that this is a glass flask and it's hot, so make sure you use one of these little hot mitts when you pick it up. And then you're going to pull it out and swirl it a little bit and kind of fold it up to the light. And you can see that it still looks pretty opaque. So you're going to keep microwaving it for 15 to 30 seconds at a time until it's completely clear.
That actually looks. That probably is okay.
Yeah, good enough. Okay, so now we're going to let it sit until it's cool enough to touch. So once your flask is cool enough that you can touch it without burning yourself, then the agarose is probably cool enough that you can pour it. So the first thing that you need to do is add ethidium bromide, and that's the fluorophore that's going to allow you to visualize the DNA after you run your gel. And ethidium bromide is a carcinogen, so we need to do this carefully, and that's why there's this special ethidium bromide station that's kind of marked over here in the back of the lab. And you want to make sure that any ethidium bromide work that you do stays on this bench paper here. There's a special pipette and a special tip eject thing that are devoted to ethidium bromide. So, Jackie?
Alright, so all you really have to do is just add the appropriate amount of ethidium bromide. And then just swirl it to mix.
Next we need to assemble the kind of thing that's going to hold the gel while it's solidifying. And so you have this tray, plastic tray, and then 2 of these black bumpers. So we can each do one. You're going to take, the bumpers have this little cutout here where the end of the tray slides into, and it's kind of tricky to get them in, but they should sort of slide in somehow. There we go. So you'll put a black bumper on each end of the tray. And then you're also going to need to put one of these ladders in there. So this is what's going to create the wells where you'll actually load your samples. And you want to think at this point about how many samples you need and make sure that you use an appropriate comb. So I'm just going to throw that one in there. And there are these little notches in the tray where the combs fit into. And you want to use the top one, Jackie. There you go. So once this is ready and your agarose is cool and you've added the ethidium bromide, then you're going to pour. So go ahead. And you don't have to measure out 25 mils per gel, just about half of the solution. And if you watch as you pour it, you should make sure that the liquid comes above the level of the bottom of the comb, so you're going to make sure that you actually create wells.
You also want to avoid bubbles.
And if you do get any bubbles, the easiest thing is to just take a sterile pipette tip and just kind of sweep them off to the side or away from the comb so that they don't mess up the wells. And then you're going to wait until it solidifies. Okay, once you think your gel is solidified, you're going to pull the comb out and pull the bumpers off. Okay, so what just happened when she pulled the lower bumper off is the wells kind of split apart. So you can see it's now torn all the way along here, and you don't have wells anymore to run. So that's something that happens sometimes. It's more common if the gel is not completely solid, so you probably could have let this solidify for a little bit longer. And the other thing you want to just make sure you do is every time you pull up the comb or pull off a bumper, just go slowly and be careful that you're not kind of just yanking the whole thing apart. But we have another gel, so we can try again. Once your gel is solidified, you can tell because it should look pretty white and opaque. And you can kind of wiggle it around and it doesn't move at all.
So the first thing you have to do is take out the comb, and you basically just kind of grab it in the middle and pull straight up evenly, and then it'll just pop out. Next you have to take off the bumpers. It's easier to take off the one that's farthest away from the wells first, and just kind of slowly work from one corner and just kind of pull it off. And do the other one. And then you want to put it into your apparatus. Make sure that your wells are closest to the black electrode, because the solution, the buffer is going to run from the black to the red. It's going to run to red, so kind of think of it that way. And you have to add your buffer next. So just add a little bit to each side. And then you actually want the buffer to cover up your gel so that it's completely submerged in liquid.
And we only have one gel in here right now, but you can put 2 gels in here. So in the lab, there'll be 2 gels for the 4 students at each pod.
So next we'll load the gel. And you just add about 5 to 10 microliters of solution to each well.
And you want to be careful when you load your samples. So one thing Jackie just did is put something dark underneath where the wells are, and that can help you see the wells. And then you want to put the pipette tip down into the well, but not so far that you actually pierce the agarose gel.
So next, when all the gels are loaded, you'll put the cover on and match up the black and the red. So make sure the red is on the bottom. And then you just plug it in.